on some conversation starters. So first off, do you guys want to introduce your teams and your projects?
Yeah, so I can go for Stony Brook University. So my name is Anika. I am here representing Stony Brook University. We are located in Long Island, New York. And our project is making an optogenetic kill switch in order to mitigate the effects of gene flow.
All right, so my name's Gabe. We're from University of Rochester, up a little north in New York. And our project is developing a new diagnostic test for endometriosis, which is a uterine disease.
Okay. So a question for both teams. So I'll be asking some questions, and we could have some back and forth about, what each team is doing for their project or how they're coping with certain, challenges. So my first question is, what is the overall impact your team is aiming to make with your project?
I can try and answer this one. So endometriosis is like a really underdiagnosed disease. It's a huge issue that our team noticed. It's a really common disease. About 10% of women have it. But at least in the US, the current statistic is that, it can take an average of like 10 years to get diagnosed with it because there's just not enough information. So our team wanted to create sort of an easier, simple diagnostic that's really cheap, really fast. And in that way, you can help detect endometriosis earlier and like stop all these women having to deal with it for, a decade of their life without any idea of what's happening.
Awesome. I think it would definitely help a lot of women as well.
I definitely agree with that. So for Stony Brook University, while we were deciding early on last spring what we wanted to do with our project, we had a lot of going back and forth. But what it came down to is that we realized the stigma around genetically modified organisms in food, especially. Like when you bring up GMOs in food, people just kind of run away from you. So we actually we wanted to focus on the scientific effects of GMOs. Though when we say GMO, people just run away. There's actually reasons why scientists believe that GMOs are bad, and one of those reasons is gene flow. So we wanted to combat this issue of gene flow to ensure that genetically modified organisms in food would be safer to use. So we first started off wanting to implement our project in vertical farms. So basically any vertical farm could grow genetically modified food in their farm because it's a very controlled environment. So they can modify the GMO and watch it while it's growing. But say that the genetically modified organism is accidentally somehow escapes that controlled setting. We wanted to make it so that if it escaped that setting, it would just stop growth on its own. You wouldn't need any pesticides, herbicides, any chemicals or anything to go out and go kill that GMO. It would just, on its own, it would be programmed and it would just stop growth. We now want to actually implement our project in both vertical farming and greenhouses because they're both types of indoor farming, and indoor farming all typically uses the same kind of growth mechanism. So we want to spread our project from now to vertical farming to greenhouses as well.
Sounds like a very useful application, and definitely good effort to investigate more about what we don't know about GMOs. Awesome. So another question for both teams. What is or are the main methodologies that have been used in your project, and how has this changed or affected the way you view synthetic biology?
Yeah, so obviously it's been challenging with COVID because there was so little we could actually do. So for most of the semester, we were really just doing literature research and just trying to figure out what is available out there and what can we possibly do. But then once we identified a huge part of our synthetic biology is making synthetic antibodies for our test. So we've really just been looking at what are the existing antibodies, how do they work, how can we, put them into a plasmid, but then also how do we get all of the effective parts of an antibody without making it overly complex, because obviously, it's a very challenging thing to try and synthesize antibodies in like E. coli. So it's really just trying to figure out how can you integrate every little piece of an antibody into a plasmid and then have it construct itself. But unfortunately, because we don't really have lab access, it's difficult to really like go through that engineering process of evaluating and seeing how it works and then refining our process, which is unfortunate.
Yeah, I would agree. For Stony, sorry. Can you guys hear me?
Yes.
Okay. So I would agree that's like very similar to like Stony Brook. We also didn't have access to wet lab. So in terms of like proof of concept and, the traditional experimentation we do, that wasn't really available to us. So we also kind of had to switch to this heavy design aspect and into like molecular dynamic simulations as well. And so I think the really positive spin on that is we kind of really have to deep dive into literature and see what worked and what didn't for our mechanism. So the original optogenetic protein we were going to use, we thought we had it all figured out. We had like this whole design built up, and then we realized after meeting with a couple of professors that there were two problems. Is one, you would have to supply exogenously the chromophore, which is a light sensitive region. And, if you have a plant that's escaping environment and you can't track it down and then, give it extra in case it doesn't have enough for itself to do that whole kill switch mechanism, that was a big problem. And also that we found out that we were originally going to kill the plant altogether, not just stop its development. But the problem with that is if you start killing all the leaves, it actually propagates the plant to flower more quickly and give on its, to reproduce and give on its pollen and its seeds. However, that specific plant or that crop will give off its offspring. And, that's obviously the exact opposite of what we want. That would just promote gene flow. So that got us thinking. And I think if we had done wet lab and we had just, gone in and we started working with the original optogenetic protein, we wouldn't have even considered that, okay, great, the design works, and maybe we had even made the science part of it, kind of work, but applicability-wise, it wouldn't have been as solid. So I think it let us slow down and really like nitpick a lot of those little details out.
I'm actually curious, could you guys explain like how your project works? Because I'm really curious about like what the mechanism of this is.
I can go for that. So our project is to create an optogenetic kill switch. And if you don't know what optogenetic means, it means something that's activated by light. So ideally, we want to introduce our optogenetic switch into certain plants that are grown indoors, such as, well, our model organism right now is Nicotiana benthamiana, but we want to introduce it to also more commonly grown crops such as tomatoes, corn, and rice. And how this optogenetic switch works is we have a optogenic protein, which is a light activated protein called, UVRA, I blanked for a second. UVRA, which is activated by UVB light, and its binding partner, COP1. So what happens is when UVRA and COP1 interact with each other, it's able to initiate transcription of a gene. And this particular gene that we want to transcribe is a tasiRNA, and it's going to target a gene that's commonly it's pretty much conserved in plants. It's called the Wuchel gene, and this gene is responsible for maintaining the stem cell population in the plant. So if we silence this gene using an siRNA upon activation with UVB light, then the plant won't be able to have the population of stem cells it needs to grow the leaves, the stems, the flowers, and therefore it will stop development.
That's actually really interesting. That's a really cool like signaling pathway to promote that.
Thank you.
I agree. Very like Gabe said, it's very interesting signaling pathway and very good use of silencing the gene. So if we feel free to ask questions to each other as we move along. So I know that modifying your methodology for University of Stony Brook actually revealed some things that maybe wouldn't have been noticed if it had just been in the wet lab, which is fantastic. Are there any other parts of your project that your team has found particularly challenging? I know also, Gabe, you mentioned that fine-tuning the antibody is, you know, not is quite challenging.
Yeah, so actually one of the things that's really challenging is the design we wanted for our diagnostic is a lateral flow assay, which is kind of similar to like a pregnancy test of like a paper strip with antibodies on it that will like detect your molecule as it passes. And, you know, we were trying to find like, you know, a paper that just explained, you know, how do you make a lateral flow assay? What dimensions do you need? And then we sort of, as we got into it, realized that everyone just kind of guess and checks their way through making a lateral flow assay. Like everyone just tries a bunch of different ones of different positions, different dimensions, different materials, and then just sees what works best, which was obviously really difficult for us because, you know, we can't actually do that if we don't have like a lab. So we tried to do that through modeling, and it was actually really cool because we were able to, I think, create some pretty novel models for that, which was something that we otherwise wouldn't have done if we had lab. But obviously, you know, it's still really challenging. And then I think the other big challenge we had was really just like working as a team. Because, you know, what was originally going to be us spending all day together in the same room, I think it's challenging always, and this goes, you know, for everything, not just iGEM, but keeping up communication, knowing where other people are at on their different parts, or, you know, if people are slow to respond, that can, you know, have a whole cascade of problems throughout the team. And that was something we had to really work hard on to sort of operate as a team.
Absolutely. And I mean, it's like you said, it's not how you envisioned working on this project in the first place. It's definitely a lot to get used to, people's different changing schedules and keeping up with everyone.
Mm-hmm.
Do you have you experienced anything similar University of Stony Brook? Or are there ways that you've kind of dealt with the same things and found different coping mechanisms to help with similar issues?
So we actually have a Discord specifically for iGEM, and what I really like about it is we've divided into 3 categories. There's like the main like top announcements and weekly meetings category, which is like for very important stuff. This is like everyone should check this daily. Like if you're not going to check any of the other chats, like these are the ones to check. This is like the most updates, and you know we really only put the most important stuff for that week there. And then we have a like sub channels for research, one for modeling, one for outreach and fundraising, and the whole like human practices aspect. And within like the whole outreach aspect, we even have like subcategories of our collaborations, of our education programs, our outreach with you know professionals in the field. And then we have like our fun channels. So when you know the work week is done and we kind of want to get together to play games or you know just talk for a bit, we've always had like an hour or 2 in the week where we're all just like, we miss social interaction, so we'll hop on the onto chat and just talk. And the chat feature on Discord is also really useful, just for work purposes. We can share screen just like we can on Zoom and type up and you know pin things and save them. So I think it's been a really good way to organize everything. And our team leaders, Anika and Alexis, are our 2 sub-team leaders. They've done a phenomenal job kind of keeping everyone on track. We kind of divided up our team into like sub-teams. So we have like a modeling and outreach and research sub-team. And so we have weekly meetings for all of these. And I think that's been a really good way. And we also have a web development for our wiki. So it's been a really good way to have organization and, you know, people can kind of pick and choose what they want to focus on and which channels they want to go to, which meetings they want to go to. So they feel like they're participating, but they're not overwhelmed with everyone spamming. So that's kind of how we've been running things, and I've really enjoyed it so far. It's made the whole experience really, really good.
That's really great. I think that's a really awesome way to, you know, categorize and have everyone participate, but not have them feel overwhelmed, like you said. Very great way to organize and you know, make the communication very straightforward.
Actually, sorry, I'm curious about something.
Sure.
Because this is just something that like I've only had my perspective on, so I would love to hear another teams. But like when you guys are doing work, like is it more like you do independently and then you come back together and compare each other's work, or do you guys like do a lot of work together? Like you're in a call together, going through stuff.
I can take that. So for us, it's like I feel like it's a combination of both. Most of the time we do work independently, but we do collaborate on a lot of things. So like if we have a collaboration that we're working on for another team, like a journal article that we're writing up, then we work together. We hop on the Discord call, and then we popcorn style when we read, and we, you know, ask questions to each other when we're reading scientific papers, for example. And then there are other times when we kind of delegate, the sub-team leaders delegate tasks, and you're expected to do those tasks and have them ready for next week. And we also have weekly meetings. So we had a big, like, 14-person weekly meeting on Tuesdays where we kind of catch everyone up for people who aren't in those particular sub-teams. But we also have sub-team meetings that are more specific for like research or outreach and fundraising and web design, so that people who are interested in those and helping parts of those projects, they can participate in those meetings and speak about what they contributed this week to the project.
It's very similar to like how we're operating right now.
And that's really cool to hear.
So I'd definitely like to add to that. This year, to present your project, there is a project video being presented. So how is that coming along for both of your teams?
I can take this one for Stony Brook. So basically, we just apply what we've been doing. When we see a task, we like to divide our labor up, get ideas so that, you know, you're not just stuck on one idea. We have more than one idea, and then we reconvene and talk about our ideas. So actually last week, it was our last meeting for the summer. This week is the first week of university. We started classes back up again. So last week we divided the team up into 3 groups, and we're all like coming up with our ideas to see what we want to see on the video. And then each team is like now thinking of ideas, and we're gonna meet actually later today as a team to discuss the ideas we've came up with.
Yeah, we've definitely been like gearing up for that presentation. I mean, everything in the team is like, I think as we work on our wiki, we're kind of letting that inform our presentation. So we've recently started like our professor, our like head advisor reached out to someone in the university that's in our like writing department. And so we've been having workshops with her to sort of figure out how to write effectively. And as we do that, you know, that helps us figure out what are we going to put in our presentation, because, you know, your wiki and your presentation should really match each other. And then we've also just been like getting experience building that sort of things up with like, we participated in like an iGEM symposium a couple of weeks ago where we had to do, you know, a similar like 15-minute presentation on all aspects of our project. You know, a lot of times we'll do meetups or collaborations and we'll make presentations. So it kind of feels like we've been building up for this, and now it's just kind of like, you know, really finalizing it and then figuring out all the different aspects and how to make it as clean as possible before we actually record it.
Okay. And I imagine that it's just maybe for both teams, it sounds like it'll probably be a compilation from each person's work rather than the traditional video inside the lab that might showcase the work differently.
Yeah, definitely. But I think also a challenge that we found is, at least for our team, we realized that all of our sub teams were kind of working too independently, and it became a huge issue of when we asked each person, like, can you sum up the whole project? Like, everyone just had aspects of the project that they just weren't well informed enough about, because they were never on that sub team, so they didn't know the sort of the reasons behind everything they did, the rationale, the research. So I think that was something we realized that we had to work really hard on, is to get every team member to understand every part of the project.
I see. Great. So, overall, you guys have discussed some of the good things that have come out of changing your methodology, and things that might not have come to light if you were in the lab. And but overall, your iGEM experience is definitely different than previous years with the pandemic. But overall, how has your iGEM experience? How would you say your iGEM experience has been so far? Having to make all these modifications and being in a completely unique experience.
Oh, you okay? Um, I mean, I think while originally when we had to start going online, I was like really, really bummed out about it, because I was really looking forward to the unique experience of working with 11 other students in a lab every day for the entire summer. But I think it's proved to be a really unique opportunity to really test how like us as students can drive this project. Our advisors tend to be very hands off. Like, they really want this to be a student driven experience, which I appreciate, because I think it sort of forced us to have that experience that you never really get in school, where it's very much structured, very much someone telling you what to do, and we've had to figure out, okay, have a problem, right? Or we're lacking in this area. How can we get together and solve this problem? And I think that's given me like a lot of experience and a lot of really good information that is super applicable to any sort of career I'm going to have later in life. And I think that's been a really unique opportunity that I otherwise don't know if I would have had.
Yeah, I definitely agree with Gabe. When I signed up for iGEM, I thought that I would be spending my summer in a lab learning things and doing things and, like with the team all the time and coming in and like, I expected it to be a summer that I would spend on campus basically and just live there. But once everything got shut down, I don't know, it was a bit of a sad moment because we weren't all ready for this. But I think it allowed me to find my niche in iGEM. Like, I expected to do lab for some reason, and I don't know why I expected to do that, but I didn't even end up doing anything research or design related. I ended up, being the outreach sub team leader and like doing outreach stuff, and I ended up loving it. And I ended up picking up something for like web dev, and I also ended up loving that. I never ever would have thought that I'd like learn HTML and CSS ever. For some reason, I thought that I'd be learning how to work in a lab and like all that stuff. So although I was really bummed out when everything closed down, at the end of the summer, I learned so many new things that I never thought that I was going to learn. So I'm really grateful for that.
That's really awesome. It's really great, Gabe, that you really feel like you got such a great professional and academic gain that with skills that you're going to use forever. And Anika, also you discovered skills that you didn't, that otherwise you may not have, because of what happened and the situation, and how you had to respond. Alexis and Julia, did you want to add to how your experience, how this iGEM experience has been for you?
I could say pretty much the same as Gabe and Anika. I signed up for iGEM, think that I would be spending a majority of my time in the wet lab doing wet lab experiments, because that's primarily what I was interested in. And even though I didn't end up doing that, I spent a lot of my summer reading articles and refining our design, get down the nitty gritty details of our design, making sure that it'll definitely work our system. And it's allowed me to think in a much more critical manner, and also to really be like force myself to do like sometimes it's kind of difficult waking up in the morning thinking, oh, I'm gonna spend like my whole day just staring at a screen, but I learned to like, you know, be like motivate myself.
Definitely.
I would say definitely, like, I'm pretty on par with Alexis. So I definitely came in thinking wet lab. I couldn't wait to get my hands on a pipette again, because I had done research beforehand in high school, and this was like my first college experience, and I was really, really excited to get back at it. But what I really did appreciate was some of the things that are a big part of research that you don't really realize, because you're not really given the opportunity to do those things, like the grant proposal writing. We're doing a journal collab and like writing up an article, and it's been really fun to do those things. That's like a part of research, and you know, really widely done for people who are in the field and, you know, kind of higher up and have had experience with it. But as undergrad, you don't really get to touch those things. I know I definitely didn't when I was, you know, in the lab with my pet, and I might have been done experience, and like I might have been running experiments, and I was doing, you know, the lab work, but I didn't get to participate in, you know, kind of creation of the project and kind of doing those things like writing. And you know, I enjoy talking about the things I've found and sharing that with other people. So I think that for me has been the positive experience, that it's so cool that, you know, I got to do those things as an undergrad, because I don't think you usually are allowed to, or you know, that's not usually assigned to you. So
That's really great. I'm glad you had those opportunities with this project. So everyone